Okay, good morning, everyone. It's 8:03, so we're going to get started. Welcome to the Research Coordination Network webinar series focused on junior researchers studying SARS-CoV-2 in wastewater. This webinar series is meant to celebrate everyone's service during the pandemic and learn from each other in a more formal format than through the Slack messaging and other informal interactions. We continue to look for people to give webinars. So if you have a student, postdoc, or you are an assistant professor or would like to nominate someone, please send an email to me or Jill. Although we do have the schedule quite full, I think, through the spring. Today we have two speakers. We have Josh Steele, who works at SCWRP, which is my favorite acronym. It stands for Southern California Coastal Water Research Project. And then we have Nishita D'Souza, who is a postdoc at Michigan State. Josh will be going first. And Josh, you can go ahead and share your screen while I talk about how we do Q&A. So Q&A is done through the Q&A chat box, not the chat. So don't use the chat, use the Q&A. Please post your questions there. And you can upvote questions that you like and you would really like the moderator to ask the speaker. The attendees do not have the ability to speak during the webinar, but we are able to enable that in situations where we think it might be appropriate. So please put your questions for the speaker in the Q&A. So with no further ado, Josh, you can go ahead and begin.
All right, thanks, Allie. Thanks for having me come and speak to you guys about some of the work we've been doing with wastewater-based epidemiology and measuring SARS coronavirus in sewage. So we, as just a bit of background, we're big fans of wastewater epidemiology. It's a great idea, and it's been working fairly well for tracking population levels recently of SARS coronavirus for the recent pandemic. It's worked in the past for things like poliovirus and other waterborne diseases fairly well too, and it doesn't have the same drawbacks of individual testing, such as the delays that it takes to get people tested once they're symptomatic and all of the things that you're all well aware of. California has been one of the states that has been charging ahead, promoting the concept and trying to figure out the best ways to use it. There are more than 35 wastewater treatment plants that have been collecting samples since the pandemic really got going last spring. More than a dozen research programs, many publications, many by folks that are a part of this group. And the CDC has recently chosen California as one of the pilot locations to try and actually use the wastewater monitoring as a public health tool and sort of combine the data with the public health data. And to that end, the State Water Board and the Department of Public Health is partnering recently with 5 utilities in California. They've started it in 8 different states, and in California, 4 of the 5 are the Southern California utilities that are member agencies of SCWRP. So we are right in the mix with all of this, and wanted to figure out ways that we could sort of add to the general knowledge and science. The emphasis, understandably, has been on speed. So, everyone's kind of used their pet method, or maybe whatever method they started with, to measure the virus. Differences in the methods do lead to some problems comparing results, compatibility across regions, across plants, and there have been a few very well done comparison studies. But even those, which there aren't very many, the WRF study is the one I'm thinking of in particular, has showed wide variability up to 2 or 3 orders of magnitude across methods. And these kinds of differences can make a big difference when you're getting at the limits of sensitivity. So especially once we're getting to, hopefully, lower levels of transmission after this recent wave. Or if you're moving into smaller sewer sheds or trying to track smaller outbreaks, which is hopefully what we'll be using this for in the near future, rather than sort of the widespread use we've been doing now. So we thought one of the things that we could jump in and do is to investigate some of these sources of variability. Basically, our plan was we would try to identify as many sources of variability that would affect method sensitivity as we could, and use controlled experiments to see if we could see what the effect of some of these different method choices make. And then we would want to apply these methods for our collection and processing. So I'm going to go through some of these different sources of variability and the methods we've tested, and then show you in the end sort of what we've seen recently in some of our monitoring. So we sat down and sort of tried to look at all the different steps and where the variability could come in, and we came up with this general flow chart, which has a lot of sources of variability on it. Everything from the type of sample you're taking, influent or settled solids, whether you're looking at your sampling strategy, your sampling frequency, how you're processing or preserving the samples, whether you're inactivating the viruses and how you're capturing the viruses, how you're extracting the RNA, which genes you're targeting, and then even the measurement method itself. I'm not going to be able to cover all of this in 20 minutes, so I'm going to focus today on just a few of these steps, in particular the processing and preserving by either refrigeration or freezing, virus inactivation by heat, and also the virus capture, direct processing, direct extracting, basically direct processing versus concentrating on a filter first, and then which gene target that you select, focusing on N1 and N2 in particular. And for most of this talk, for all of this talk, I'm going to be talking about influent samples, not settled solids. All right, so I'm going to jump right in. For the frozen sample preservation, we realized this was an issue, and because we had a lot of plants that in the beginning collected their samples, both influent and settled solids, and then didn't really know what to do with them. So they immediately froze them at -80 if they had a -80 freezer, and figured they'd figure out what to do with them later. Those of us that have worked with viruses, especially RNA viruses, we know that some of them you go through 1 or 2 freeze thaws and they just disappear on you. So we were a little worried and wanted to actually take a close look at how this affected these viruses in particular. So we just basically set up a simple experiment where we took influent from 3 different plants and processed these samples in triplicate to try and get a sense of the variability within the plant. And then we stored one set at 4 degrees, and then we froze a set of the water at -80 degrees. And then each of these, subset of those, we did direct extractions with no concentration, so just taking the influent itself right into an extraction kit and going from there, or we concentrated on a 0.45 micron HA filter with acidification and magnesium chloride. So what did we find? Well, the direct extraction, it looked like that freezing. So you have the black here is the fresh processed samples, and the red are the frozen samples, where the water was frozen and then processed. What we found was that the direct extraction, it seemed to actually make a pretty big difference. What you're comparing here, you have the plant 1, 2, 3, and then plant 1, 2, 3. So you're sort of comparing the first dot in each set, second dot in each set, third dot in each set. So in every single case, we saw a decrease in the concentration of the coronavirus once you've frozen the sample for the direct extraction. Curiously, we did not find that to be as big a decrease when we did the concentration on the HA filter. This honestly kind of surprised us. So we are looking a little more into that. These are fairly recent results, but at this point, it looks like you should avoid freeze thaws when you can, especially if you're doing direct extraction of influent. But it's possible if you have frozen samples. For now, it looks like it's possible that you might be able to use a concentration method like the HA concentration method, HA filtration, to actually be able to use those archived samples. One of the next things we looked at was virus deactivation. a lot of the clinical labs and our lab were required to have a biosafety level 2+, or the clinical labs themselves, if they're have the live virus, have to have a biosafety level 3. But many of the labs of the sewage treatment plants or other folks who were processing just don't have that level of that biosafety level. And getting that level actually would require some pretty substantial modifications that they weren't willing or weren't able to do. So one of the things folks came up with was heat treatment, pasteurization, to make sure that the virus is inactivated before you do any processing, before they even come into the come out of their sample bottles and are processed in the lab. Now, I know there has been some evidence, particularly the folks in New York have looked at with PEG precipitation and found that there is either no effect or even a benefit to pasteurization. But we didn't know that we hadn't seen anyone do that with sort of the methods we were focusing on, the direct extraction and the HA concentration. So again, similar design. We had influent from 3 plants, processed in triplicate, kept one set at 4 degrees, and then did our normal processing. The others were pasteurized upon arrival at 70 degrees for 40 minutes, which is one of the pasteurizations that we had heard folks were trying, and then did process them the same way. In this case, we found that in both the direct extraction and the HA filtration, we saw a pretty substantial decrease, 1 to 3 log reduction, about 1.5 log reduction on average with the direct extraction, and about 2.5 to 3 with the HA filtration. So from the samples we had and the way we process them, pasteurization does make a substantial difference. If you're down near that lower level of detection, you might lose your signal entirely if you pasteurize your samples. Moving on to virus capture. So we were just curious, what were the benefits or trade-offs? We knew, folks like LA County SAN, and we're doing no concentration at all, just directly. That's the LA County Sanitation District. They were just taking Influent, putting it into the extraction kit and extracting it and going from there. And other folks had liked that. It's a way of chemically inactivating the virus quickly. We also had traditionally found a lot of success with concentrating on HA filters with other viruses. So that was with both acidification and magnesium chloride. And there had been the publication showing that also worked fairly well with this virus too. So we wanted to test these. What were sort of the benefits? What were the drawbacks to doing these different ways? So here you can see we have filtration on the left, direct extraction through a magnetic bead kit, the BioMaru kit, and then a direct extraction through the Zymo kit. And their kit, this kit is the one that LA County Sanitation Department was using. And what you can see is they actually work fairly comparably in terms of the results they're getting. So when you filter a larger volume, but when you convert it to a per 100 milliliter concentration, you actually get fairly similar results, especially with the same kit. The Zymo kit does look to be a little bit more sensitive. However, what we did find is when you get low enough results, especially with the SARS coronavirus, so with the bovine coronavirus on the top here, our spike in control, you can see that you get very comparable results across plants, comparable results across 2 of these methods. The Zymo kit does a little bit better at recovery. When you look at the SARS coronavirus, which had a lower concentration by a few orders of magnitude, this is where you see a difference. The sensitivity in the direct extraction, you start to lose. And so these down here at 0 are actually below the detection limit on the assay we were using. So while you get maybe better recovery with the Zymo kit, when you have higher concentrations, you can certainly, and similar recovery with the BioMaru, the magnetic bead kit, you lose sensitivity. We can detect lower levels, mostly I think because of the larger volume we can capture on the HA filter, even though it's at a lower recovery efficiency. So finally, the gene targets we took a look at. This was sort of early on in the pandemic when we started thinking about this, because there were multiple primer sets available, but labs didn't really have a choice to, they tended to pick one and run with it. They didn't tend to take a lot of time to worry about which one they were picking. They just picked one. And recently, this monitoring in California that has been started with the CDC in partnership with the CDC, they focus solely on the N1 gene. But a lot of our long-term data sets, at least in Southern California, we have both. So we kind of wanted to take a look at N1 versus N2 and how are they similar? Are they different? Are they telling us something different? As a quick reminder, these are all the different primer sets. The N is the nucleocapsid gene, which should be fairly conserved, which is why they have a bunch of primer sets for that. Some of the others we have not yet tested, but there are still others that you can take a look at or use, and people could go back into archive samples and try and see if they like those better. So when we take a look at N2 and N1, and this is just some of that monitoring data that was started more recently for the CDC, you can see that the N2 does track the N1 pretty well, at least in this plant. This is from the Hyperion sewage treatment plant in Los Angeles. It tracks fairly well with N1, although it is pretty consistently the same concentration or higher. However, when you sort of do a scatter plot of them, plot them together, they correlate really, really strongly. So you could argue that either one is fine. It's really hard to tell if one is actually the truth or not. I mean, in some cases, when the values are high, you do get a decent spread between the two. When we look at all the plants over the entire time we've looked, we actually get a similar result. We get a slightly higher number of N2 copies compared to N1 in most cases, but they're highly correlated to one another. This is across multiple plants in each of these counties. It's really unclear to us which gene is true, and so one of the things we've decided to do, and a few other plants have too, is when we report it, we tend to average the two. Okay, so I've given you guys a lot of our methods that we took a look at and what we thought affected things that didn't affect things. And so this is sort of what we came to for our monitoring samples. We took our composite samples and held them at 4 degrees, made sure to process them within 72 hours. Usually we're faster than that, but that's the longest we let them go. We concentrate 20 milliliters of Influent. We amend it with magnesium chloride and adjust the pH to 3.5 or lower on a 0.45 micron HA filter, which is a mixed cellulose ester filter, and spike in bovine coronavirus as a sample processing control. We do bead beating lysis and extract using the magnetic bead kit. We also put in an extraction inhibition control. And then we measure N1 and N2. We measure pepper mild monovirus and our controls with digital PCR. We convert this concentration that we measure to a sample concentration, like a per liter or per 100 mils, depending on who we're reporting to. Then we adjust it for recovery. So far, we have not normalized it to the pepper mild monovirus. That's something we are still looking into as well. So what does it look like when we do that? Well, here we have, going back to April, this is our sort of time series of average N1 and N2 in millions of gene copies per liter. And you can see, and then down here we have Los Angeles County cases. So this is from Hyperion treatment plant on the top, which is the city of Los Angeles, one of the larger treatment plants in the state. And I believe in the world. And here on the bottom are the daily case counts. And you can see that they track fairly well. For what it's worth, if we had switched methods, when we started doing the monitoring, that would have been over here right at the beginning of December. So what we decided to do was sort of stay with what we'd done, do it all the same way, so we could compare all the way back to April. And when we do this comparison, just a really straightforward scatter plot and correlation, we find that we have a pretty good relationship between the coronavirus concentration and the cases in Los Angeles County. A 0.78 correlation, Bonferroni corrected p-value of less than 0.001. So we are pretty pleased with even just a really straightforward comparison using these methods, this concentration method, to the case numbers we're seeing. So in summary, methods matter, and the devil is in the details. You can change your outcome, even not detect things that are there, because some of the method choices can change things by more than 2 orders of magnitude. We generally recommend against changing methods over the course of monitoring, but if you do, you have to be very careful when you're comparing across methods and make sure you understand exactly what the differences are doing. Our other recommendation is when virus concentrations are low, you may need to switch to more sensitive methods. If you are not concentrating viruses, you may have to start. And including controls and sample processing controls can help you compare across these methods. And we do have other sources of variability, as you saw from our big flow chart that we're addressing. But I had 20 minutes, so I chose these. I thought these were pretty good ones to show you for today. And with that, I just want to acknowledge the public utilities agencies that helped us with collecting samples and also did some of the research on the methods as well, especially Los Angeles County Sanitation District, City of Los Angeles Sanitation, Orange County Sanitation District, and City of San Diego Public Utilities. I want to thank all the people at SCWRP who did a lot of this work along in collaboration with me. It's always a group effort. And I also want to thank Ali Baim, Deneen Blackwood, Rachel Novo, Sandra McKellen, Shu-Chen Fang, who have had lots of conversations with us since April and doing all of these, and were very helpful and helped us think very deeply about some of this. And I'm happy to take any questions.
Great, thank you so much, Josh. That was an excellent presentation. Just as a reminder, if you have any questions for Josh, please add them to the Q&A box, not the chat. We'll look at the questions in the Q&A primarily, and you can also upvote questions if you want to see them answered faster. So I'm going to just go ahead and start up at the top. One question coming in is in relation to the data being shared between among the California utilities. How is that data shared between the 4 utilities that you mentioned? And are there any plans to publicize it in a dashboard or other method?
That's a really good question. Right now, so there's 3 basic answers to that question. The first is, it is right now, it's sort of shared at the will of the agencies themselves. So some of them have been very public with it, some of them haven't. That is going to change right now. The sort of the partnership between the public health and the utilities that's been started since December, they are collecting data on a more, under a more tight schedule. And so that should be able to be public soon. I believe that's being put into both the CDC database and a state database. As far as a dashboard, though, that I don't know of yet.
Okay, great, thanks. The next question is about your assays. So how big of an issue is self-complementarity or primer-dimers on concentration? Wondering if assay design issues might be affecting the N1 and N2 assays.
I think that is something that could be affecting them. We did some optimization in the beginning of those assays where we looked, and we do actually multiplex the 2 of them, so we run them together. We did not find, we were able to optimize them at a fairly reasonably high annealing temperature, and I'm gonna get it wrong if I say exactly what it is now. So if you want to know, I'm happy to share it with you when I have it, when I can make sure I check it. But we haven't seen a huge amount of primer dimer issues or those kinds of issues coming up in our data that we've looked at, or at least not enough to substantially change the results.
Okay, thanks. Our next question, it's a little bit of a comment, but we can definitely, this is a good one. So I mean, you recognize that some people have had different results, right?
Absolutely.
With respect to freezing and fresh or pasteurization and not. And so wondering, you showed data for 3 samples across those comparisons. Have you evaluated it on a broader basis? So basically, do you have a sense of whether or not the results you're seeing are really consistent with your methods? And if you want to comment on maybe some of the other factors that might be influencing your results there.
Sure. I mean, so I want to emphasize again that this was with Influent. So, liquid samples, basically water samples with solids in them. We didn't remove the solids from the raw Influent. We tried to keep them suspended. So this is a specific sample type. I think if you're doing a different type of concentrating, like a PEG precipitation, or even a larger volume concentration, these results might well be different. I mean, we were surprised with the freeze thaws, especially that even concentrating 20 mils seemed to make a substantial difference in how much that freeze thaw was affecting things. So yeah, I think absolutely there is variability between the sample type and the method, and so it's something that basically we're encouraging folks to take a close look at and make sure that they're getting the result, that it's not affecting their results.
Thanks. One other question about what's your detection limit on your assays? I know you mentioned a couple of tests where it came up below that limit.
Right. So let's see, on a per liter basis, our detection limit is typically around 2,000-ish copies for the HA concentration method. So, a couple copies per mil. And the detection limit is lower when we do the direct extraction by about, probably about tenfold. Higher by about tenfold. So that we lose our detection limit about tenfold higher when we're looking at the direct extraction.
Okay, thanks.
Mm-hmm.
And I think we have one question left, and maybe just enough time for it if we answer quickly. So do you know if people in California are taking an action based on poor recovery or severely inhibited samples? Like, are they rerunning or anything like that?
I know that we adjust for recovery when it's above a certain threshold, and if we don't have that threshold, we rerun them. I can't speak to everybody, but for the samples we're rerunning, that's the case. I believe for at least some of the other treatment utilities, they do rerun them as well.
Great, thank you. All right, I think that will finish up our Q&A. Thank you so much, Josh. Before we go to our next presenter, I just want to really quickly make a quick announcement from the RCN. We just recently posted a call for mini proposals from students. We're looking to host a series of videos that can train about methods related to SARS-CoV-2, wastewater surveillance research, lab methods, analysis methods, whatever you want. So I'm going to post the webpage link into the chat, and we'd appreciate if you would share that information with any students in your networks that might be interested. And the details are there, and so is my email address if there are any questions about that submission process. So I'll add that to the chat, and with that, I think we'll turn things over to our next presenter, Nishita D'Souza. You can go ahead and unmute and share your screen.
Thank you. Thank you for this opportunity to share some of our results from Michigan. I'm a postdoc working with Dr. Joan Rose at Michigan State University. And today I'm going to be sharing results that from our methods optimization studies and monitoring in Michigan. So just to give you a brief timeline in terms of the coronavirus cases in Michigan, although the first case was detected in the US in January 2020, Michigan saw its first case in March 2020, and ever since then we've had about 600,000 cases of the COVID-19 in the state. Recently, in January 2021, we saw our first case of the UK variant, and currently we have about 61 confirmed cases related to that particular strain in the state. So looking at trends of the virus cases in the state, we saw that there have been three waves in general, one starting up in the beginning of the pandemic between March and May. And then the second big wave was seen between October and December. And we also saw a small wave starting up in January, and now are beginning to see a decline in the cases that are associated with the virus. Coming to our workflow, we have been collecting samples from wastewater treatment plants in the state, as well as monitoring the Michigan State University campus and the surrounding neighborhoods. We've been collecting grab samples initially and have moved to composite sampling. We've been testing influence and effluence, and I'll be sharing some of those results along with some information on our methods. So for our influent sample processing, we use 100 ml of our wastewater sample, which we concentrate using a PEG precipitation method. We further centrifuge the sample and recover the pellet, which is then further RNA extracted and quantified using droplet digital PCR. We have been looking at the N1, N2, and E targets since the beginning of the pandemic, but of late in January, we dropped the E target, and the N1 and N2 seem to track pretty well over most of our samples, which is why we continue to process using those two targets. For our effluent samples, we have been concentrating 1 liter of wastewater sample and using the InnovaPrep concentrating pipette to concentrate that high volume, and then we further elute it using the elution canisters provided with the equipment, and RNA extract and quantify using droplet digital PCR in a similar manner. Now, in terms of our workflow optimizations, in the beginning of the pandemic, we did test 3 different methods, 2 ultrafiltration methods and 1 PEG centrifugation-based method. We used 5-6 to assess recoveries. 5-6 is a Pseudomonas enveloped phage. And what we found using the 5-6 for recoveries is that the PEG had significantly higher recovery percentage as compared to the ultrafiltration methods. And given the considerations of cost, the issues with supply chains, etc., and the high recovery, we decided to go ahead and process our samples using the PEG method. Now, one of the bottlenecks with using the PEG method was that the method involved a 16-hour hold period post the addition of the PEG, and that was increasing our processing time. So we decided to look and investigate if we were to remove that 16-hour hold period, if we were able to get similar recovery. So when we did test that, we noticed that although the recovery did drop, with the removal of that particular step, the recovery drop was not significant, and now we continue to process our samples without that drop. Therefore, we are able to process our samples much faster now and actually deliver results on the same day if required. Well, we participated in an interlaboratory study with the Water Research Foundation. We were one of the 32 labs that used this particular method. So 4 of the labs used the method that I just described. The other labs used different methods. And what you can see is these 4 labs, in terms of their N1 and N2 targets, performed pretty well in terms of the actual concentrations across the board. and there was also reproducibility between these particular labs. We also looked at OC43 recoveries from these particular plants, and once again, if you see, these 4 labs had good reproducibility across with the same methods that were employed. Since we were using 5-6 in our particular lab to assess recoveries, what we also did was a comparison between the OC43 and the 5-6 surrogate, and what we found was that the recoveries that we were estimating using 5-6 were comparable to the OC43 recoveries, and therefore we continued to use 5-6 in the lab. Now, there's been a lot of talk about whether we should be testing grab samples, composite samples. If grab samples, what time those grabs should be taken, and if composite samples, how many hours should we be actually sampling for. So we did a study which compared grab samples and composite samples, grabs taken at peak flow at 10 a.m., and then 7-hour composites and 17-hour composites. And what we saw across all 3 gene targets is that the concentrations were not very different with this particular exercise. So this is good news for utilities which don't have automatic composite samplers. They can definitely take targeted grab samples which would be indicative of the concentrations, as well as being able to use 7-hour composites is helpful in being able to get the samples into the lab faster and getting results out at a rapid pace. We also looked at the effects of freeze thaws on samples. So what we did was a study with raw wastewater that was analyzed initially when it came into the lab, and then we froze a portion of that wastewater at minus 80 degrees C for varying periods of time. And what we saw was after the one freeze thaw cycle, we saw a reduction in the concentration of the virus about 58 to 70% across the gene targets. So this is not a favorable way of storing the samples. As far as possible, it would be ideal to be processing the samples as soon as they come in. Now, based on our workflow, we were also curious to see that if it was better to freeze the concentrates that we got post PEG versus freezing the actual raw influent samples. So we also did a study to see if there was any decrease in terms of the concentrations when the concentrates were frozen. However, what we did notice was there was no major decrease in the concentrations. And in some of the samples, we did see a slight increase in the RNA. So overall, the goal would be to try and process the samples and then freeze the concentrates, and then these concentrates, the concentrations would remain stable for quite a long period of time, which could then be used later to do further analysis. We are also now a part of the Michigan Wastewater Surveillance Network, which is funded by the state of Michigan. So this is a consortium of about 18 labs that have been monitoring the wastewater in Michigan through a pilot project between October and December. So the sites in blue are the sites that have been monitored by these particular labs, and they've been using the method that I described for the influent samples. And the sites in orange are the actual testing facilities. So I'm going to share some of the results from our testing over this period of time. So this is one of the wastewater treatment plants that we were sampling higher population. And what we noticed was that in the beginning, when we started sampling in April 2020, we were seeing the signal bounce around a little bit in the population. And then once restrictions were imposed in the communities, we saw that there was a drop in signal. And then further, once things started reopening, we started seeing a high incidence of cases in these communities, and also an increase in the wastewater signal post September. Similar trend was seen in this wastewater treatment facility also, again, around 100,000 in population being served by this particular facility. And we saw a similar increase in October after a consistent signal right from July when we started monitoring, but there was a sustained higher signal between October and December. Coming to one of our smaller wastewater treatment facilities here, although when we first started sampling in the beginning of the pandemic, we did see a few peaks. There was a consistent non-detect result that we saw for samples over a period of many weeks. And then in September, we started seeing an increase in the concentration, and that went up pretty high in October and sustained right through December, when we started seeing it drop to a certain extent. Similar trends seen at this particular location also with a smaller population size. So clearly the population size is not really affecting the way that we can look at these trends in the communities. And most of the cases, the wastewater signal in the communities are tracking well with the cases. This is one of our other communities that we've been monitoring, which includes residential facilities as well as some other entertainment-based facilities. So here we were monitoring with grab samples. So you can see that there has been a slight, bouncing up and down of the signal and a lot of non-detects initially. But later on in the pandemic, we started seeing these increased concentrations. And now in some of the facilities, we're actually seeing the concentrations drop, which is a good sign. At this particular area, we also measured the concentrations in the influent basin and the effluents that were coming out of that particular plant. And as you see, as the influents have been increasing in concentration, we did start to get detects in the effluents that we were measuring later on. So this is something that we will further explore in terms of whether it would be beneficial to be testing the effluents at this particular plant. So our preliminary findings suggest that the concentrations in the wastewater appear to track well with the cases in the state of Michigan. So this particular exercise could inform clinical testing and could be a leading indicator of cases in the community. The low concentrations or no signal was detected during periods of the stay-at-home order and when restrictions were imposed in the state. And so we believe that these particular results could inform on public policy in controlling the spread of infections in the community. I would also like to share some results with regards to our MSU wastewater sanitary investigations. So we've been looking at a total of 11 sites over the last year or so that we've been testing. Initially, when we started testing, we were looking at 4 different sites. Typically, we saw that on campus, because there were no students in the dormitories, there weren't very high concentrations. However, in the nearby communities, we did see that the concentrations were increasing along with the increase in cases. And we did notice one particular peak in the monitoring initially when we were monitoring in June, which was associated with a sample that we took on the 15th of June. And on the 23rd of June, an outbreak was identified in the community, which was linked to an East Lansing bar event. And we believe that this peak was indicative of that particular outbreak. And so we feel like this shows promise in providing an early warning signal for these particular kinds of outbreaks in the community. Coming to our campus wastewater monitoring exercises. So in the fall, when a few students started coming back to campus, we started monitoring the dorms extensively. And we did 4 particular dorms on a weekly basis. And we were, at this point, we were collecting grab samples from the dorms. So some of the, concentrations that we did find, even with the grab samples, were pretty high. But overall, since there were only about 3,000 students on campus, we took this more like a pilot study to learn what we can do with these kind of exercises. And we were able to detect a high concentration through some of the samples. So the data would suggest that we can see the wastewater signal increase about a week prior to the onset of symptoms in the students clinically. We do feel like we missed a few of the peaks because we were collecting just grab samples in the mornings, and moving ahead, then we did take the decision to move towards composite sampling. We also, based on these results, felt that maybe early infections did excrete higher levels of the virus. And this is something that we are currently exploring with our spring semester monitoring. So these are some of the results from the spring semester monitoring. The first week that you can see here is the week that the students were just moving in, so not much activity in terms of high wastewater signals. And then as the students continued to trickle in, we started seeing the development of higher signals. And then about 2 weeks into the students moving in, some of the dorms did show very high levels in the wastewater, which then subsequently with quarantining and isolation, those levels have started coming down at these particular dorms. So this kind of reporting has really helped the administration, combine their clinical efforts as well as the spit testing efforts that are ongoing on campus, and we'll be looking at doing further correlation with this data going ahead. So what's next in terms of what we are going to be monitoring? And I want to leave you with some kind of few questions to think about. So can we be using this information as a leading indicator and as an early warning sign for cases developing in the community? And that's something that we have seen few glimpses of promise in terms of the data. Will the information that we are collecting help support rapid testing programs, taking into account the wastewater data as more of a screening tactic to decide where to test more clinically? Would demonstrating relationships between existing diagnostic testing methods and wastewater surveillance data be useful? So we are further exploring these correlations and looking to see if we can actually draw this kind of positive relationship between the two. Are there any specific areas of concern or vulnerabilities on which leadership would like to focus? So whether that may be schools or colleges or long-term care facilities, correctional facilities, what is the exact focus for this kind of investigation? Would risk mapping be a useful output? So would looking at risk analysis and cases and wastewater data together provide models that would be useful to explore further incidences of the outbreaks in the population? And lastly, would sequencing and tracking new variants be useful to map the virus transmission in the community? And this is something that we are looking at in terms of our old archive samples as well as the samples that we are currently testing. So with this, I'd like to thank all the members of the ROSE team and everyone who's helped out with the study, Michigan State University Infrastructure and Planning, and the State of Michigan Department of Environment, Great Lakes and Energy, and all the Michigan wastewater treatment plants that were participating in the study. I'll be happy to answer any questions.
Thank you. That was an excellent presentation. There's a lot of work there at every step, so I'm really impressed. So the first question we have in the Q&A box is if, and I noticed that E did track pretty well with N1 and N2, and so we're curious, or an audience member was curious why you dropped the E target, and if you have tried looking at the S target.
Yeah, so the E target was dropped partially also because it was more expensive to run the E target. The E was tracking well with the N1 and N2. We are currently running an N1 and N2 duplexed assay. So we're able to run one assay and get similar results. The E concentrations also tended to be lower, and we were getting a few non-detects. particularly with our sanitary sewer samples, because the concentrations are much lower in the sanitary sewer samples. So we felt it would make more sense to, pay more attention to the N1 and N2 targets. Also considering for wastewater utilities that the cost would increase if we were doing multiple targets. With regards to the S gene, we are now looking at that, considering that a lot of the variations are mutations linked to that particular gene. So we've started testing some of our samples with that, as well as looking at mutant detection for the variants. And our results are very preliminary at this point. I've run maybe 50 odd samples, but the S gene does seem to be tracking well. I'll honestly have to run more samples in order to comment more on that.
Awesome. One question I had, just based on the number of sewage treatment, the number of treatment plants you're monitoring, if you've noticed any trends with respect to, for example, combined versus separate sewers, and whether or not that impacts how well the clinical maps with the wastewater signal, or are there any trends you've identified looking at kind of the sewage system parameters and your data?
So each of these systems that we've been monitoring are very different. And to be honest, we haven't, dived really deep into looking at those correlations with the cases yet. But having said that, we have seen that some of these peaks at some of the communities are very distinct to those particular communities. And hopefully, the pilot data from the Michigan network is going to soon be looked at more intricately. So once we do look at the data from around the state, we'll have a better idea of trends within the different communities.
So on the pilot data side, similar to the previous presenter, does Michigan plan to share more of the data on some sort of publicly available interface or dashboard in the future?
Yeah, so the state of Michigan currently does have a site. So some of the images that I did show you were from that particular site, wherein currently there are just the testing sites and the sampling sites that are uploaded there, but we do plan to have the data available to anyone who would need to access it and have that kind of information for the work.
Okay, and then similarly on your campus surveillance, is the data how is the data shared with students? Is the data shared with the students?
So currently the data is being shared with the administration. We haven't been doing extensive monitoring before the spring semester. Right now the clinical case information is being shared with the students on the website, and we do upload the data to a dashboard, but the university is still working on making that accessible to the general public at large.
So this is a question from the box. I guess, have you done any work, in addition to all the work you've done, have you done any work on surface waters or being able to detect the virus in surface water to link it to the wastewater results you've gotten, or anyone else in your department?
We've been testing largely wastewater. We do work with microbial source tracking, and surface waters is something that we do look at in our lab. However, we haven't really tried to link any of that kind of work to the SARS-CoV-2 work that we've done with wastewater.
Okay. So yeah, this was kind of an interesting thing. Early on, you showed the grab versus composite data, and it looked like they were pretty consistent. But in the wastewater treatment plant, but maybe in the sewers for the dorm, you guys are moving towards composite sampling. So maybe if you could comment on that discrepancy.
So when the data that I did show was for the wastewater treatment plant, wherein there is much more mixing and things are more uniform. However, in the sewer shed, we did notice that initially, since we were taking grabs, there is that hit and miss chance of, missing the actual signal moving through the sewer shed. So we did want to make sure that we were able to grab as much of the information as possible. So we decided to move to 7R composites in the sewer shed, just to ensure that we're able to get as much information as possible. So, and we believe that the 7R sampling is working really well for us, and that also helps us get the samples into the lab, sometime in the afternoon, and then we're able to report results very fast with that versus trying to do 24-hour composite samples.
Okay. So I guess this could go to either presenter, if either of you want to comment. You have, we determine limited detections for our assays, and someone is asking about how that might relate to prevalence, disease prevalence, and if there's a way to relate our limit of detection to disease prevalence, incidence rate per X number of inhabitants. I think that's what this question's getting at.
I mean, I can jump in and say that sounds like a really cool idea. And I think it would be a really interesting thing to try to do. I think depending on the size of the sewer shed that you're dealing with, that could become really tricky. You might be able to get it as far as an average level, but when you're dealing with a really large sewer shed, it might become hard to nail it down to convert that to it. Not that it can't be done; it's just going to not be a trivial calculation.
Yeah, I think a lot of people have tried to do this, and I think based on your presentation, you kind of showed all the steps where there is uncertainty, and so being able to back that out would require probably a lot more time to understand that uncertainty. Well, I think that is the last questions, unless Nishita, you really want to comment on that last question. I think we will conclude this webinar, and thank you both for your presentations. They were excellent.
Thank you.
Thanks.
Thank you.